Hello, everyone. This is the video for problem set number 11. Our topic today is prokaryotic gene regulation. We're going to start off with some general information about operons and general vocabulary, and then we will look at gene regulation using the operons, the lac operon and the trip operon, which are both found in E. coli. So, when we talk about operons, there are two parts. The first word is either positive or negative control. Positive control tells you there's some activator that's present, and the activator binds to the DNA to help initiate transcription. Negative control tells you there's a repressor that's present, and the repressor binds to the DNA to prevent transcription. That first word, positive or negative, is always something binding to the DNA, and then positive, we get an increase in transcription, negative, we get a decrease. The second word is either inducible or repressible. And I think about this as the small molecule that interacts with the molecule that was binding to the DNA. So this second molecule is not binding directly to the DNA, it's binding to either our activator or our repressor. When we have inducible control, when the two molecules bind, then that means the inducer is our small molecule, it's present, and when that binds, it can cause transcription to occur. When we have repressible control, when our two molecules are present and our co-repressor binds, it causes transcription to turn off. So let's look at the positive and negative just a little bit more. In positive control, remember, our regulatory molecule binds to the DNA to turn on transcription. Negative control, our regulatory molecule binds to the DNA to turn off transcription. And inducible and repressible, our second molecule, the effector molecule, typically it's a small molecule and it's going to interact with our regulatory molecule. So positive or negative, we're looking at a regulatory molecule. Inducible, repressible, we're looking at an effector molecule. Regulatory molecules bind to the DNA. Effector molecules bind to regulatory molecules. And in an inducible system, when the inducer binds to the regulatory molecule, it turns on. So inducer turns on transcription. I use TX as my abbreviation for transcription. A repressible molecule, in a repressible system, the effector molecule is often called the co-repressor. It's going to bind to the regulatory molecule. So the effector binds regulatory molecule, but it turns transcription off. Okay, so that's some general comparison of a couple of those terms. Let's see if we can look at some examples. So I'm not going to do every one of these for you. I'm going to pick a few to do and then let you handle the rest on your own. You can look up the key and have answer if you have any questions. So let's look at the first one. Transcription occurs because the regulatory gene product binds to the DNA. So remember, Whenever the regulatory protein binds to the DNA and causes transcription to occur, we have positive control. So that would be positive inducible and positive repressible. I guess the part I kind of left off on that last page was you can put positive inducible, positive repressible, negative inducible, negative repressible. So we can use those first two words is in all sorts of combinations. That means when the regulatory molecule binds to the DNA, it turns off. We must have negative control. That's the whole definition of negative control. Regulatory molecule binds to DNA, turns it off. So that would be both negative inducible and negative repressible. I'm going to skip the trip and the lac operons because we will be going over those in a bit more detail. So transcription occurs with a high effector level. Remember on our last page, we talked about inducible control, and that would be when you have a high effector and that causes transcription to be on. So both of these with inducible control would be answers here. Transcription occurs with the absence of the effector or low effector, and that would be repressible. Because typically when you have the effector present in a repressible situation, that's turning transcription off. And then the regulatory gene product binds to the DNA in the absence of effector molecules. That happens with negative inducible and positive repressible. And the regulatory gene product binds to the DNA in complex with effector molecules. That happens with positive inducible and negative repressible. And I think these last two will make a little bit more sense as we talk a bit about the lac and the trip operons. So let's look at the lac operon. The lac operon's goal is to break down lactose in the cell. So it's providing the enzymes necessary to bring lactose into the cell and break it down. And it doesn't need to be on all the time. If the cell doesn't need the products that lactose is broken down into, glucose and galactose, don't need to make those enzymes. And if there's no lactose present in the media, the cell doesn't need to make those enzymes. So when you think about it, and we're all kind of lazy, even E. coli. So E. coli doesn't want to make those enzymes unless it needs those enzymes. And the lac operon and the regulation of that lac operon help that to be done efficiently. So in this first diagram, let me see if I can make each of these just a bit larger. I know they'll blur, but maybe they're easier to see too. Okay, so in the lac operon, outside of the operon, we have a regulatory gene product that makes the repressor. It has its own promoter, so it has its own control. But our lac operon is actually this part here. It's got the promoter region, an operator region, and then three structural genes, LAC-Z, LAC-Y, and LAC-A. LAC-Z makes the enzyme beta-galactosidase, LAC-Y makes the enzyme permease, and LAC-A makes transacetylase, and we're not quite sure what transacetylase does. LAC-Z actually splits the lactose into glucose and galactose, and because that's what beta-galactosidase does, and then permease helps the lactose to get into the cell. We also have RNA polymerase needed in order to transcribe and translate those genes. This is a polycistronic messenger RNA product that is formed when the lac operon is on. So the way this works is the regulatory gene makes a repressor. If there's no lactose present, that repressor binds up here to the operator region. And that if the repressor is bound there, RNA polymerase can't bind. Think of that as a roadblock. And that means there's no transcription. And I'll just write that a little bigger, no TX for no transcription. And this is in a situation with no lactose. But over here, lactose is present. That means there's a little bit of allolactose present as well, and the allolactose binds with the repressor. that changes the conformation of the repressor. Without allolactose, like this, with it like this. Once the repressor changes conformation, it cannot bind to the operator. That means RNA polymerase binds to the operator. It goes all the way down, transcribes, makes messenger RNA, and then we have translation of the three structural gene products, beta-galactosidase, permease, and transacetylase. So that's the general idea. So let's look at this a bit more, and I'm going to have to shrink this down now so we can see both the questions and the reactions at the same time. Okay, so RNA polymerase, where does that bind? Bound to this area here, which is called the P region, and P stands for promoter. The repressor binds to the region that kind of overlaps with the promoter and that first structural gene. It's called the operator. Now, we do have various mutations of the lac operon. I'm really hoping that you already have watched the regular lecture video on this. The OC mutation, that is a constitutive mutation. It means there's a bad operator region. And if there's a bad operator region, we don't get the repressor binding, because the repressor, in order to bind to the operator, it has to fit precisely. And if that operator is mutant, they won't fit together. So that means the operator cannot bind to the repressor. And that means we have what we call constitutive production of the gene products. So we can say transcription occurs. The IS mutation, this is a mutation of the repressor. Normally, remember, the repressor had the top binding site that would bind to the operator and the bottom binding site that binds to allolactose. This is a mutation that affects that bottom binding site. So the repressor cannot bind to allolactose. So that means the repressor is bound to the operator all the time, so no transcription. So all of the operons in the cell would be turned off. Let's look at a few genotypes. Now, in your lecture, we went through a few single mutation genotypes, and then some of these that are basically partial diploid bacteria. You might remember the F factor from our lecture on conjugation. Think about one of these strands of DNA being on the bacterial chromosome and that second strand being on a plasmid or another piece of DNA that's in that bacteria, so that we have a diploid. We have two copies in this particular bacterial cell. And the question is, for each of these, is beta-galactosidase produced or not? And is permease produced or not? And we want to use these three words. Inducible means that when lactose is present, it is produced. When lactose is absent, it is not. Constitutive means that that enzyme is produced all the time. Not expressed means it's not produced at all. So we're going to use these three words in the blanks. Our first lac operon, we have normal repressors, I+ on both of these, so we have a good repressor on both. This first one has a bad promoter, and because of that, none of the gene products on that piece of DNA can be made because this bad promoter can't bind RNA polymerase. The second one has an OC, and that OC means it's a constitutive operator. It can't bind to the repressor, so it's always on, and it has a good promoter, but it has a Z-, so beta-galactosidase is not expressed. Even though the operon is on, since that's a bad gene for beta-galactosidase, we don't make beta-galactosidase. It has a Y+, and since that is a constitutive operator, it is produced constitutively. So I'm going to let you do Well, actually, I'll go ahead and do them. So this next one, IS versus I plus. When we think about what's dominant and what's recessive, IS is dominant to I plus. And we know that because when we have this IS, that's going to cause that repressor to stick to all of the operators in the cell, effectively turning off both of the operons. So not expressed for either of the two gene products. Remember, the repressor acts in a trans manner, which means it can affect both pieces of DNA in the cell. And it does that because it's a protein. It goes and floats all over the cell. Something like a promoter and an operator are cis acting. They only are going to affect that same piece of DNA. Now we have I plus and I minus. I plus is dominant to I minus. And then we have on this first gene a good promoter and a good operator and a good beta-galactosidase gene. On the second one, and we have a bad permease gene. On the second one, we also have a good promoter and a good operator, bad beta-galactosidase gene, but a good permease gene. So since we have a good repressor, that repressor will bind to both of these O plus normal operators when lactose is absent, but will not bind when lactose is present. So in both of these cases, we will get inducible control of both enzymes. Let's see, inducible. So for both enzymes, they will be under inducible control. Notice that this first piece of DNA only makes good beta-galactosidase. The second one only makes good permease. But when we look at that cell, since it's got both of those pieces of DNA in it all together, both of those gene products are made inducibly. It says the portion of the lac operon shown in the problems above, these problems indicate which aspect of control, and that's negative inducible control. Negative control, repressor binds to the DNA to turn off gene expression. Inducible, allolactose binds to the repressor to allow transcription to occur. Well, there's a little bit more to this lac operon. We also have something called catabolite repression. If we think of breaking down lactose, it gets broken down by the enzyme beta-galactosidase into galactose and glucose. And then galactose actually is two glucoses hooked. It can get broken down further somewhere else. But the point is, if the cell already has lots of glucose, it does not need to break down lactose to make more. These two are the catabolites, or the breakdown products from this reaction. Catabolite repression occurs when these breakdown products, in this case, glucose, goes back and regulates the amount of this reaction that will occur. Which by that, I mean, if there's a lot of glucose in the cell, having a lot of glucose will tend to prevent lactose being broken down, even if lactose is present. And it does that because there's another aspect of control here. It's an aspect of positive control. Typically, there is a cap cyclic AMP complex. Cap is our protein that binds to the DNA, and cyclic AMP is our small effector molecule. If cap and cyclic AMP are joined together, they bind to a site, and that allows RNA polymerase to bind effectively. So here we can see cap and cyclic AMP together binding, and it's called a cap binding site, and that allows RNA polymerase to bind well and transcription occurs. However, and this happens when there's not much glucose present, and when there's not much glucose present, there's a lot of cyclic AMP. But there's a little loop where if glucose is in high concentration, it prevents cyclic AMP from being formed. So if glucose is high, we don't have much cyclic AMP. And even though we have CAP, the catabolite activator protein, it cannot bind by itself, which means RNA polymerase really doesn't bind well, and transcription does not occur. This is actually positive inducible control. Positive control, CAP binds to the DNA. Inducible, our effector molecule is cyclic AMP. It binds to CAP to allow it to bind to the DNA to cause transcription to occur. So all of these things tend to get interrelated in a cell, and that's typical of cell physiology. Lots of processes are interrelated. Let's switch gears and look at the trip operon. The purpose of the lac operon was to break lactose down into its component parts. The purpose of the trip operon is to synthesize or produce tryptophan. And there are a series of 5 enzymes needed in order to make tryptophan. There are, by the way, the lecture has some really good videos of the lac operon and the trip operon, and I highly encourage you to watch those because you can kind of see how this flows a little better than my still diagrams can do. So the idea is there is also a repressor involved, but the repressor cannot bind by itself. In order for the repressor to bind to the operator region, it has to complex, and it complexes with the effector molecule tryptophan. So tryptophan is our co-repressor. It is considered our effector molecule here, and that is our repressor. When these two are complexed together, they bind to the operator, and there's no transcription. Well, that makes sense. Why would the cell make more tryptophan if it already has a lot of tryptophan present? And so if there's a lot of tryptophan, the tryptophan binds to the repressor, the complex binds here, turns this off. This is called a negative repressible system. Negative, repressor binds to turn off DNA, binds to the DNA to turn off transcription. Repressible, the tryptophan or the effective molecule binds to the repressor to help it turn off transcription. But when there's not much tryptophan, the cell's going to need tryptophan. Tryptophan is one of our 20 amino acids, so when the cell makes proteins, it will need tryptophan to make those proteins. So the repressor does not bind by itself. It's not bound to tryptophan because tryptophan is low. RNA polymerase binds to the promoter. It goes down here and it transcribes all of those structural genes, and translation occurs, and we get all of those gene products so that tryptophan can be produced. And there's actually a precursor called chorismate. You'll hear that mentioned in the videos, that's converted through these enzymatic steps to tryptophan. The trip operon, though, has another sneaky little part to it. A man named Yanofsky figured this out. He was looking at mutations in the trip operon, and he noticed that his point mutations, he could map them in order, and he could see a collinearity between the DNA and the proteins. And so if he had mutations in this order, his amino acids in the proteins would be in a collinear manner. So Yanofsky was famous for working with the trip operon and noticing the collinearity between DNA and protein. He also figured out this unique aspect in the trip operon called attenuation. So when there's not tryptophan present, sometimes when there is tryptophan present, sometimes, even though this is blocked, RNA polymerase manages to go ahead and start. So we don't want transcription to occur here. Yanofsky figured out that there is actually an area of messenger RNA produced before that first gene product will need to be translated. And within that area, if he had deletions in that area, he noticed he got a lot more transcription and then translation of his enzymes than he would have thought when there was plenty of tryptophan present. So he looked at that area a bit. And he noticed that this area has 2 codons for tryptophan in a row. Now, we know bacteria are pretty cool things. We know you can do a lot in bacteria. One cell membrane, no nuclear membrane. So transcription occurs, translation occurs. In fact, if this line right here is my messenger RNA, I've got RNA polymerase binding going across, and right after it, I've got my ribosome going across. So transcription and translation are occurring simultaneously. If tryptophan is low, we want those gene products produced. And if tryptophan is low in the cell, here's RNA polymerase, it's going along. Here's my ribosome. RNA polymerase gets to the point where the ribosome is, where the ribosome is going to need to find transfer RNA that carries tryptophan. So it's going to pause at those 2 codons, and RNA polymerase gets way far ahead. And as the messenger RNA is formed, sometimes we can make hairpins in the messenger RNA because bases kind of pair together within that one RNA chain, and this is the hairpin that's formed. This is not a termination hairpin. Remember when we talked about transcription in prokaryotes, we said that there is a sequence where you get a hairpin formation, and then that kind of cues RNA polymerase, we're going to end transcription. Well, when tryptophan is high, RNA polymerase is chugging along and the ribosome is right behind it. They stay close together because the ribosome does not have to wait for charge transfer RNA with tryptophan because there's lots of tryptophan. And when that happens, the sequence of the RNA forms this termination hairpin. And that causes termination of transcription in situations where we shouldn't have transcription anyway. But somehow this is a situation where RNA polymerase managed to jump over and got in here. So think of this as an extra control on transcription. So thinking about the strip operon, let's see if we can figure out this chart versus high versus low tryptophan. So what we're really doing is we're looking at those top diagrams there and get them both on the same page, sort of. Okay, almost. Okay, I think you can see those diagrams in the chart now. So with high tryptophan, it's down here, is the trip repressor bound to tryptophan? Yeah, it is. Is the trip repressor bound to the operator? Yes, it is. And we'll look at this termination bit in a minute so we can look at the other diagram. With low tryptophan, it's up here, is the trip repressor bound to tryptophan? No, because there's no tryptophan to bind it to. And is the repressor bound to the operator? Also no. Now let's look at this other diagram. Is the anti-terminator, that would be this one, is the anti-terminator formed in the messenger RNA? With high tryptophan? Nope. With high tryptophan, we get the terminator hairpin formed. And is transcription attenuated with high tryptophan? Yeah. This is high tryptophan. With low tryptophan, is the anti-terminator hairpin formed? Yep. Is transcription terminated? No. As I said, I really encourage you to watch the videos on this. This is tricky. It's complex regulation, and it helps to watch those videos multiple times. The videos are in the lecture, and there's also a link on Moodle to them. Please contact me if you have any questions, and thank you for your attention to this video. Have a great day.